Cassidy.
And my name is Jenna.
And we're going to talk to you about type 4 glioblastoma.
Let's begin by giving an introduction of the disease, and later we'll explain some research that's being done to help find a cure for it.
Glioblastoma multiforme, also known as GBM, is a type of brain cancer.
Glioma is a type of tumor that forms in the brain and spinal cord, beginning its growth in supportive cells.
Types of gliomas include astrocytomas, ependymomas, and oligodendrogliomas.
Glioblastoma is most commonly found in the frontal and temporal lobes of the cerebral hemisphere.
Now that you know the basics of what GBM is, some of you might be wondering if you're at risk for developing it.
The disease occurs randomly. However, there are certain factors that increase the risk of developing a tumor, including ionizing radiation to the brain or spinal cord, or the use of harmful chemicals.
Fortunately, if you have a family history of brain cancer, there's little known genetic or hereditary influences.
Overall, the risk of developing glioblastoma increases as one ages, the average being between 45 and 70 years old, and the median being about 64 years old.
In addition, the disease is more commonly found to affect males.
Depending on the location of the tumor, the ways in which GBM affects the nervous system vary, but overall it's rare for the tumor to spread outside of the brain.
Symptoms arise either from the tumor itself or from the fluid surrounding the tumor, which results in increased pressure and swelling in the brain.
Some common symptoms include headaches, vomiting or nausea, loss of appetite, and double or blurred vision.
Other symptoms affect neurological functions, for example, changes in mood and personality, or the ability to think and learn, along with difficulty in speech, the occurrence of seizures, sensory changes of the face, difficulties in balance, and memory loss.
Unfortunately, once diagnosed, there is no known cure that completely eradicates the tumor while maintaining normal function of the brain, and glioblastoma usually ends in death soon after diagnosis.
There are, however, some techniques that are able to slow down the growth of the tumor and delay the onset of more severe symptoms.
The first step is usually surgery.
It's extraordinarily difficult to remove the entire tumor without damaging the surrounding brain tissue, making the realistic goal to remove as much of the tumor as possible.
By debulking the tumor, there is a better chance that further techniques such as radiation and/or chemotherapy will be able to kill the weaker exterior parts of the tumor.
Unfortunately, radiation and chemotherapy can only slow growth of the tumor and cannot completely remove it, usually resulting in the death of the patient within 11 to 15 months.
However, we read about some research being done in an attempt to find a cure.
One example is the identification of a small molecule that's part of the signal transduction pathway which induces apoptosis in GBM cancer stem cells, better known as GBM-CSCs.
Here we're going to talk about the methods used and the results found through this experimental process. This figure shows that the metabolite of RAP-GBM called cRAP-GBM induces apoptosis in GBM-CSCs by interacting with RIPK2.
This may sound very confusing, but don't worry, we'll explain everything. The selective apoptosis-inducing molecule that was found is known as RAP-GBM, which stands for receptor interacting protein kinase, glioblastoma multiforme.
The molecular form of RAP-GBM is shown on the left.
After interacting with the receptor, it works as a kinase by phosphorylating, or adding a phosphate group, to the molecule that comes next in the signal transduction pathway.
Through the metabolite identification method, RAP-GBM was converted in vitro into a different species of that molecule named RAP-GBM18, or cRAP-GBM, which is basically just the cyclic version of the initial molecule RAP-GBM.
The molecular form of cRAP-GBM is shown on the right.
Part B of Figure 2 shows the conversion between the normal and cyclic versions of RAP-GBM.
Researchers compared different species of RAP-GBM, both the normal and the cyclic, and tested them in different cultured cells, some diseased and some not. The diseased cells having actually come from the tumors of diagnosed patients.
The researchers found that there was more conversion of RAP-GBM to its cyclic version in diseased cells.
The rate at which conversion occurred was found to increase even more with the addition of the reducing agent NABH4 to the medium.
The difference in the conversion from RAP-GBM to cRAP-GBM between different types of cells can be seen in the graph through the difference in colors.
There's more red, or conversion to the cyclic version of the cell, on the graph for GBM-CSCs, which is on the left, which is the graph for diseased cells, compared to the graph on the right, which is cells from the lung. This graph compares how different cells respond to an increase in the concentration of cRAP-GBM.
By showing the change in percent survival between specific cells, including GBM cells, it's clear that these diseased cells react differently to cRAP-GBM than other cells do.
you can see that the GBM cells, or the red line, die off first as the concentration of CRIP-GBM increases, which is later explained to be the case because CRIP-GBM induces apoptosis in GBM cells.
This is a photo of the CRIP-GBM PAP, the PAP part standing for Photoactivatable Affinity Probe.
This molecule was designed and synthesized so that researchers would better be able to visualize how RIP-GBM interacts with the next molecule in the signal transduction pathway and to figure out what that next molecule is.
Through this process, they were able to identify CRIP-GBM's target to be a protein called receptor interacting serine threonine kinase 2, better known as RIPK2.
Part D of Figure 2 shows the structure of the PAP molecule that played a vital role in discovering this important receptor protein.
Part E of Figure 2 shows that CRIP-GBM induces caspase activity, but the non-cyclic version does not.
The cyclic version does this through activating RIPK2.
The structure of the protein RIPK2 can be seen in Part F of Figure 2.
Finally, Part G of Figure 2 shows in what ways caspase is activated and how RIPK2 is expressed.
DMSO, which is basically the control group, hardly reduces the activation of caspase.
With the addition of CRIP-GBM, however, there's a lot more caspase activation, adding evidence to the hypothesis of the role of this molecule in the transduction pathway that has to do with tumor growth.
Alright, moving on to Figure 3.
Overall, this figure shows that CRIP-GBM activates caspase signaling in GBM CSCs by regulating the interaction with RIPK2 and TAC1 and between RIPK2 and caspase-1.
Part A is evidence that CRIP-GBM has an effect on cell division by showing how it reacts with different proteins in the pathway.
Over the course of 24 hours, CRIP-GBM induces the cleavage of different caspases, including caspase 1, 9, 7, and PARP, which is poly-ADP ribose polymerase.
Part B shows CRIP-GBM's proapoptotic characteristics by showing the percent survival of certain proteins when introduced to the cyclic molecule.
When interacting with the vehicle group, DMSO, it's clear that with the increasing concentration of CRIP-GBM, there's a decrease in percent survival of GBM CSCs.
When introduced with a different protein, however, there is a different response.
Researchers tested an inhibitor called pan-caspase inhibitor ZVAD-FMK, and they found that this protein did indeed inhibit CRIP-GBM from inducing apoptosis in the cancer cells.
Part C is showing that caspase-1 activation is upstream of caspase 9, 7, and PARP because pre-treatment with caspase-1 inhibitor ACY-VAD-CHO blocks caspase-9 cleavage.
The binding between CRIP-GBM to this receptor, RIPK2, triggers a molecular switch, resulting in not only increasing the formation of the proapoptotic RIPK2 caspase-1 complex, but also by reducing the formation of the pro-survival RIPK2 TAC1 complex, by decreasing the interaction between the two.
Part D of this figure shows the results from the experiment that had been conducted to determine whether or not the treatment of CRP-GBM changes CIAP1 and or CIAP2 GBM CSCs. These
proteins had previously been shown to promote the RIPK2 ubiquitination of other cells.
In the experiment, as the concentration of CRIP-GBM increases from 0 to 500, it's clear that there was a decrease in the binding between RIPK2 and CIAP1 and 2, providing evidence that these ligases in some way regulate RIPK2's ubiquitination in cancer cells.
Part E of Figure 3 shows the difference in how TAC1 and RIPK2 complexes react when in the presence of CRIP-GBM compared to how they normally react.
The treatment of CRIP-GBM greatly reduced the amount of TAC1 in cancer cells.
Since the TAC1 complex is pro-survival, CRIP-GBM is clearly of help because it's inhibiting the cell division by limiting the activation of TAC1.
This figure shows the amount of TAC1 left after co-immunoprecipitation of it.
Co-immunoprecipitation is basically isolating a protein and getting it out of a solution by using an antibody that binds to that protein.
By using an anti-RIPK2 antibody, the researchers were able to isolate TAC1 and quantify the amount of it after having been treated with and without CRIP-GBM.
The graph shows a lower amount of TAC1 present when CRIP-GBM is present, proving the relationship between these two compounds.
Part G of Figure 3 is very similar to Part E of the same figure. However, instead of comparing how CRIP-GBM affects the RIPK2 TAC1 complex, it shows how that same molecule affects the RIPK2 caspase-1 complex.
In the graph, we can see a huge increase in the amount of caspase-1 when it's treated with CRIP-GBM compared to when it's treated with the vehicle molecule.
This makes sense and agrees with the concept that CRIP-GBM induces apoptosis in GBM-CSCs because, since the RIPK2 caspase-1 complex is pro-apoptotic, if there is an increase in the quantity of that complex, there will be an increase in apoptosis.
Part H of Figure 3 relates to Part G of that figure in the same way that Part F relates to Part E.
By using the same co-immunoprecipitation technique, the researchers were able to isolate and calculate the amount of caspase-1 in the cells before and after the introduction of CRIP-GBM into the solution.
There being more caspase-1 when in contact with CRIP-GBM compared to the amount that there is with the vehicle shows again how CRIP-GBM could be a potential cure to GBM by increasing the amount of pro-apoptotic complexes.
Part I of Figure 3 shows the proposed mechanism by which CRIP-GBM induces apoptosis in GBM-CSCs.
This diagram shows how CRIP-GBM interacts with RIPK2 in a way that results in decreased association with TAC1 and increased association with caspase-1, which can then lead to the activation of an apoptotic signaling cascade.
After conducting all of these experiments and collecting all the research in vitro, the researchers decided to test out their idea in vivo, since the conditions vary between the two.
By using an intracranial xenograft model, the researchers were able to see how the alteration of the molecule RIP-GBM actually affect live organisms, in this case mice.
The figure basically shows that their manipulation of the molecule to its cyclic version does indeed impede upon the growth of the brain tumor.
This is the graph showing the tumor signal intensity in mice over a period of 5 weeks.
The mice that were treated with RIP-GBM, shown in blue, show a much lower tumor signal intensity than those that were treated with the vehicle, shown in red.
By using fluorescence molecular tomography imaging, the researchers were able to visualize and measure the change in the size of the tumor as treatment continued.
The stuff that appears to be white in the photos is actually the tumor.
With these pictures, it's clear that the mice given RIP-GBM have much smaller tumors than the mice treated with the vehicle.
The last section of this figure, Part C, shows H&E stains of mice brains at week 5, and the tumor is outlined in black.
You can clearly see that mice treated with RIP-GBM have much smaller tumors.
All of these experiments have proven that the molecule RIP-GBM is an important part of the signal transduction pathway that signals cell division in GBM-CSCs.
By altering the protein and converting it into its cyclic version, apoptosis was induced and cell division was limited, resulting in a successful reduction in the size of the brain tumor.
In other words, with more research, the molecule RIP-GBM might eventually lead to finding a cure to glioblastoma.